Good afternoon, everyone. Today, for Bio 110 Systematics, we will be reporting about the polyphily of Bikkia based on multilocus sequence analysis with emphasis on the endemic Bikkia philippinensis, including its conservation status.
Rubiaceae, or the coffee family. It is the fourth most diverse family among angiosperms and with members found mainly in the tropics. It is specious, which means that it is rich in species. It is highly endemic, which means that it is restricted only to a certain region. Philippine species constitute about 13% of total Rubiaceae worldwide and are believed to be candidatus. This only means that it is well known but understudied or uncultured. This is more evident in the case of the endemic Philippine Rubiaceae, wherein significant taxonomic efforts started only after the publication of an updated generic list. The Philippine Rubiaceae is also excluded in molecular phylogenetic studies due to lack of leaf material for DNA analysis. Philippines has the largest number of indigenous species and endemic genera of Rubiaceae, which are Antarestir bremek, Grignopsis myrrh, Solitariids, and Villarreal. Note that SBP refers to the species pluralis, which means multiple species, and bremek is a botanical plant name author abbreviation for botanist Cornelis Elizabethus Bremecamp. The genus Bikkia is a shrubby genus of the tribe Jocoseae from the family Rubiaceae with 20 species of shrubs, and there are 11 species occurring in New Caledonia, while the remaining are distributed from New Guinea, Philippines, the Moluccas, Micronesia, Fiji, Tonga, and New to the Wallis Islands. A molecular phylogenetic study of the tribe using two markers, the ITS and the TRNLF, stated that Bikkia is polyphyletic, forming two separate clades with a habitat and corolla shape as bases. One clade is formed by the species of the New Caledonia with campanulate corollas and are found in inland forests. The other clade consists of species with funnel-shaped corolla and are found in coastal habitats. The problem was mostly at failed to resolve the polyphily in the genus because of the limited sampling of Bikkia species. However, the endemic Bikkia philippinensis was excluded in their analysis due to the lack of plant material, and in spite of being endemic, it was not classified in any category in the national list of threatened Philippine plants. Modern systematics uses a combination of molecular markers to resolve taxonomic conflicts and to establish better resolution of phylogenetic trees. The three molecular markers used are internal transcribed spacer of region nuclear DNA, RPS16 intron, and TRNLF region of chloroplast DNA, which have been proven useful in delimiting relationships even at species level. These three multiple sequence data tests the polyphily of Bikkia as currently circumscribed, determines the phylogenetic position of the Philippine endemic Bikkia philippinensis, and assesses conservation status of Bikkia philippinensis.
Materials and methods. For the plant material, the researchers collected two samples of Bikkia philippinensis for the leaf molecular analysis. These samples were collected from Daco Island in Siargao and in Magpupungco, Pilar, Surigao del Norte. The collections were based on the type locality indicated in original publications and previous collections indicated in the herbarium specimen from different herbaria. For the DNA extraction and amplification, the researchers used the Kaigen DNEC plant minikit. This kit is used for genomic DNA extraction from plant material and for purifying DNA samples. The purified DNA samples from the Kaigen were then amplified using the polymerase chain reaction. They amplified the ITS or the internal transcribed spacer and took chloroplast DNA, the RPS16 intron, and the TRNLF region. Testing for amplicons. Amplicons are pieces of DNA or RNA that is the source or product of amplification or replication events. So for testing amplicons, they used agarose purification electrophoresis. This is used to separate charged particles such as DNA. Samples collected from the DNA extraction and amplification were subjected to 1% agarose gel electrophoresis to purify it. So the DNA samples collected from the PCR were cleaned using the QIA quick purification kit, then subjected to agarose gel electrophoresis and sent to South Korea and Germany for sequencing. DNA sequence analysis. The researchers used the codon code aligner. This is used to manually edit and forward and reverse sequences. They also use the MacLeod, which assembles sequences for alignment and excision of unnecessary bases. Aside from the DNA sequences from Germany and South Korea, additional sequences were retrieved from GenBank and post-assembled in MacLeod. These sequences were representative taxa from Chicoxii and two outgroups, Cinchona and Guetarda. The significance of adding the two outgroups was to determine character polarity. Character polarity is used to address an issue between two character traits, for example, character trait A and character trait B, to determine whether character A evolved from B or the other way around. The researchers also used the phylogenetic analysis using parsimony or the PAUP. This is a computational phylogenetics program used for inferring evolutionary trees. As the name says, it uses parsimony, and parsimony is defined as a criteria under which the optimal phylogenetic tree is the one that minimizes the amount of homoplasy. Bootstrap analysis. This is used to assess relative statistical support for identified clades. In all analyses, characters were all given equal weight and gaps were treated as missing data. So for the research, results with a bootstrap support of 86 to 100% were treated as strongly supported, 70 to 85% were treated as moderately supported, and 50 to 69% were weakly supported.
Before we start discussing the result of the study, let us first define what parsimony and phylogeny is. Parsimony and phylogeny states that the tree with the fewest common ancestors is the most likely. An example would be hypothesizing that if two species both have prominent incisor teeth, they also share a single ancestor, rather than they evolve the trait independently. Under the subheading Sequence Characteristics and Variation, 6 sequences of B. philippinensis from the 3 molecular markers are newly published here. The ITS dataset, although has the shortest matrix length, this marker yielded the highest number of parsimony informative characters, as seen on Table 1, followed by the TRNLF and the RPS16 intron. The tree shows the phylogenetic relationships within the Chiocosii. Bikkia was considered polyphyletic in its current circumscription because the New Caledonian species, labeled A in the tree, are found in a clade together with the monotypic Moririna, while the other species are found in another clade, the clade labeled as B, sister to Badusa. Moririna is here placed as sister to the New Caledonian Bikkia with strong support of 100. Moreover, the two clades of Bikkia gained more support in our combined tree, compared to that of Motley et al. The tribe Chiocosii was defined by a combination of two synapomorphic features. Number 1, the stamens inserted at the base of the corolla, and number 2, the spinulus pollen. However, stamen insertion at the base of corolla is also found in the tribe Hameliae, as seen in the phylogenetic tree. Thus, spinulus pollen should be retained as the only synapomorphy of Chiocosii species. In the combined tree, it is evident that the Bikkia philippinensis is nested within the costal Bikkia group. Similar to other Bikkia species found in this clade, Bikkia philippinensis was found to thrive in coastal environments, having solitary white flower and infundibular or funnel-shaped corollas. Furthermore, the widespread of B. tetranda, the type species of the genus, is also found in this clade.
For the proposed taxonomic solution, it was suggested that the coastal species, which in here is labeled as B, be retained as Bikkia since it includes the type species B. tetrandra, while the New Caledonian inland forest Bikkia, which in here is labeled as A, be transferred to another name since they differ with the coastal group in terms of flower morphology, wherein the New Caledonian inland forest Bikkia have campanulate corollas, whereas the coastal species have funnel-shaped corollas. Bikkia philippinensis is restricted along the coasts of Daco Island and Magpupungco, Surigao del Norte, Mindanao. On the conservation status of Bikkia philippinensis, the International Union for Conservation of Nature, or IUCN, red list categories and criteria were used to evaluate if a taxon belongs under a threatened category. Criterion C, specifically C2A1, was used to identify Bikkia philippinensis as a critically endangered species. In this criterion, a taxon must have less than 250 individuals to be considered as critically endangered. And with Bikkia philippinensis having less than 100 individuals in all areas surveyed and less than 50 mature individuals in each subpopulation, Bikkia philippinensis fits under this category. So that's it for the polyphily of Bikkia based on multilocus sequence analysis with emphasis on the endemic Bikkia philippinensis, including its conservation status. Thank you for listening.